Okay, so just to remind you, last week we worked through this uh, concept test for proposing a match model versus a mismatch model at the level of basically drawing an information graph structure for these two models. Um, Maybe it would be helpful if I just remind you what that looked like. So we basically had a mismatch model that looked like this, where we had population allele frequency. We had basically two different people represented by copy number variables, basically saying what's the genetic state of that individual. And I'm going to continue to use Greek letters here. Let's say kappa and lambda representing the copy number for that polymorphic marker in that individual. And then from each of those, we had like a microarray sampling process that would attempt to measure the genetic state of the individual via fluorescence measurement. And I think we called those X and S maybe, representing basically the actual fluorescence measurement from a sample taken from, say, a crime scene, and then fluorescence measurement from a sample taken from a suspect. And the whole point of this model, of course, is that by having two completely separate copy number or genetic state hidden variables, kappa and lambda, that are connected only by the population allele frequency theta, we're basically saying these are just independent draws from the population. There's two completely independent people drawn from our population. And in the match case, we had something, even simpler, I guess you could say, where we basically said, well, there's actually just one genetic state that's determining the whole picture here. And so basically our two observations were actually just measured, derived from that same hidden variable. Okay, so we have this simpler graph structure. Now,